Okay, so as I showed, within human brain, so MT is a part of the region that seems to respond to visual motion specifically. And then there is some correlator in after image, motion after effects. And if you look at the neuronal selectivity using single neuron recording in a monkey, then it turns out that each of the column, the vertical structure, this is a cortical surface. Within each column, they tend to cluster the neurons that respond to a particular direction of motion. Like here it's a top left, and then here it's a top right, and things like that. And then it gradually changes as the location within the area of the MT changes. And it also has some kind of tuning or selectivity to the visual motion near or far on top of that. So it's very tiny scale of the change in terms of direction and also the distance. So if you the hope is that if you stimulate very tiny part of the cortex like here, then overall you selectively stimulate the neurons, activate the neurons, which respond to the near and also more or less like towards the top left kind of direction of motion neurons. And the reason why I'm talking about the stimulation becomes clearer in the following. But before that, so MT neuron has been recorded for a long time, since like 80s and 90s, and then people have found that each particular neuron has a particular receptive field, like retinal ganglion cells. But in the case of the MT cells, it has a selective area responding to that particular motion in direction and the speed and the depth. So let's say if you find a particular neuron that responds to that upward motion in particular area, let's say to the right of your visual field, then that responds very strongly when all the dots are moving upward. And then if the proportion of the neurons going to the upward is decreasing, then the response also decreases. And for this type of the experiment, so visual motion stimuli like this has been used. It's called a random dot kinematogram. And it's a pretty important kind of stimulus to control what the parameter of the visual stimuli drives the neurons response. And then you can use this kind of computerized stimulus to map out whether one particular neuron likes which direction and the motion, and also the depth of the motion to be driving this neuron. So using this, the reason why it's important is that without any as a sort of a control situation where the researcher just recorded the neuron's response, then as a function of the strengths of the visual motion, like motion coherence, correlation, then monkey's response to make a decision, make a correct decision increases like this. And this is called psychometric function, psychometric function. As a function of the stimulus, the psychological sort of the performance increases or related, right? That's a black dot. On the other hand, if you just record the neurons, which respond sometimes like a lot, sometimes very small. So the x-axis of this figure is spikes number of the firing per neuron, and then they change the strength of the stimulus from 0.8 to 3.2 to 12.8, and then compare that with the case where they presented the motion stimulus in the opposite direction, and that's this black distribution. And then comparing black and white, and then drawing some, vertical line to say that how much proportion they are, accurately discriminating white from black. They could compute this, so-called neurometric curve. Neurometric. This is the white one. And then in the case of MT, it turns out that monkey's performance is almost the same as one of the best neurons within MT. So that's sort of, suggestive that neurons in MT is responsible for our visual motion behavior in the case of monkey. And then a more decisive kind of experiment came from this causal experiment. Okay, causal. What it means is that so monkey, as a this is sometimes a figure that is used to explain the time course of the experiment. So monkey is looking at this cross fixation point. Okay, they don't move eyes here. And then the receptive field of the particular neuron is mapped out already here. And then neurons also, like the motion direction of this, top left. And then stimulus is also presented. On top of that, when the researcher actually stimulated the neuron by passing electricity, like delivering electrical stimulation in that area where they recorded the neurons, where this neuron has because of the columnar structure, is next to another neuron that prefers the same direction, then they may or may not change the percept. And that's what exactly they try to measure. So it turns out that it does. So with the stimulation here, the monkey required less visual motion strength to say the same thing here. Okay, that's what this horizontal shift means. So previously, monkey said that, like, let's say in this case, maybe this one is easier to say. 80% of the time, when there is a 10% motion, they said that it's actually going to the top left. However, when combined with the stimulation, then they needed only 0% motion, so meaning that it's a random scramble things. But they still said that it was moving to the top left. So it seems like this stimulation of the population of neurons within MT changes the perception of motion. That's this causal intervention of stimulating the neurons to look at whether the perception changes is a powerful paradigm to study consciousness, and this is one of the first study to show that within monkey. And then recently, this kind of thing hasn't been done, for a long time in the humans, but something similar is becoming kind of plausible. So the paradigm of the epilepsy neural recording that I described in a couple of weeks ago, where the patients were implanted with the electrodes here, like this, blue things are all the electrode contact called ECOG, electrocorticogram. And these are the sort of the strip of the electrodes, put into the brain, like here, inserted from the top. And the purpose of this is to map out, the location of the brains that are useful or critical for the everyday functioning, and they needed to take out the particular part of the brain so that to remove the epilepsy without changing the sort of the social and the perceptual functioning. And this particular case of the study, Roche-Carr, they studied 3 patients who are implanted with the electrodes that are nearby the MT here. So the surface that you are seeing is a cortex of the subject B. And the subject B was beforehand this implantation presented with this moving stimulus twice, and then here is a response they saw. So the red part is a motion, visual motion, like you saw before that Buddha demonstration, right? They saw expanding and contracting things 2 seconds at a time. And then during the gray, they saw the static pattern. So they stopped, and then you don't see any kind of visual motion. And then this alternates every 16 seconds, okay? Motion 16 seconds and then stop 16 seconds. Motion 16 seconds. Then as you see, there is a roughly like 4 to 6 seconds of delay, but there is some substantial increase whenever this B sees the motion, and then goes down when they see the static figure. And this kind of a thing can be computed in terms of statistical reliability, and then find that okay, so maybe this particular part and this particular part of the brain for this patient happen to be MT. Defined functionally according to fMRI criteria. And then in this particular patient, it's very it happened to be the case, but this electrode 2 sit right within this fMRI defined MT area. So what the researcher did was to look at how this area actually responded to the moving stimuli. And unlike fMRI, this electrocorticography, ECG, can be measured at very, very fast and high temporal resolution, like 2000 hertz and so on. So they can shorten the duration of the stimulus much faster. So 2 seconds of the static stimuli and then 4 seconds of the moving stimuli. 1 second expanding, 1 second contracting, 1 second expanding, 1 second contracting. And then what they found was that the so-called high gamma range power, usually it's defined as 80 to 150 hertz component, or slower component, theta power, like 4 to 8 hertz, something like that. Within this ECG electrode, showed this selective response to the motion. In the case of the theta of this electrode number 2, it goes up for the expanding motion, but not for the contraction. And in the case of the high gamma, it in general increased, but in the beginning of this expansion, it showed a very, very strong response. And then when they looked at other electrodes, none of them basically showed this kind of pattern, only electrode number 2. And here's the further sort of the data to show that how this electrode 2 is special. So when they looked at only a high gamma activity, okay, so this is all high gamma activity, which is roughly speaking, we don't go into the detail, but high gamma power recorded by ECG is roughly the mean firing rate of the neurons underneath the electrode. And then when they look at that, then around the area, electrode 2, when the subject saw visual motion, which is this red dot, it's much higher in terms of the activity. Whereas when he saw the static one, it is lower than the average. So that's the blue one. But just going 1 centimeter above or below, you don't see that kind of clear difference here and here. Okay, so it's a highly spatially selective, and also this is also just an average of 4 seconds, and each dot is a repetition of across many different things. So unlike fMRI, it's extremely reliable and stable per individual. It's almost like 100%, decodability in a sense between the motion versus non-motion. Then the critically in this ECG experiment, they also do the brain stimulation before they determine where the epilepsy is coming from. if the part of the brain is, foci for the generating epilepsy, stimulating that the electrode generates epilepsy. That's sort of the empirically known. And to see this, correspondence clearly, I just flipped the vertical axis of this, brains of the subject B, but you can see the correspondence in the above. And then in this case, stimulation always need to be with the two electrodes. One is passing the negative current, negative charge, and then the other side is a positive. So you need always a pair. And then when they use a 1-2 pair, then what they found was that when the current passing between the electrode is like below 5 or 6 to 9 or 10, then they always pretty much elicited always the report of the visual motion. On the other hand, if it's straddling between 2 and 3 here, or 3 and 4, then unreliable report was elicited. Or when they increase the amount of stimulation from below 5 to 6 to 9, then 2, 3 also started to elicit the motion percept. So not only the stimulation response paradigm, but also stimulation phenomenology report paradigm, there was a very specific kind of correspondence between the electrode location and phenomenology. And here I want to also share you what the subject reported when he was stimulated with this electrode. So I think it's this one. And it's not probably easy to hear, I think, but let's see. Can you hear the sound?
I saw it.
What?
I saw you move.
Can you hear the sound? Okay, all right. So this is subject A, whose data I haven't shown, but she had some kind of an electrode around the MT. And when she was stimulated with the MT area, then a patient reported the illusion of seeing the doctor jumping to the left and back again.
I saw you move. I saw it.
What?
I saw you move that way and that way.
With your hand, show me where I moved. Approximately.
So then
At the starter.
Okay.
Here's the sham stimulation. Sham is a sort of technical term, meaning that it is stimulating other areas of the brain or not, but subjects that doesn't know or when or how, whether it was stimulated or other area is stimulated.
Tell him what you feel.
Right here.
Is he moving now?
No.
Okay.
So that's a sort of the control, condition where subject says that no, I didn't see any motion. And then this is subject B, whose data I shared readily, in depth. And then this is the report.
The top was vibrating just that corner. Okay. Now.
It's a bit hard to hear, even the original movie. So I'll just, describe what he was saying. So he's basically implanted with the left MT, as I showed in the image, and the left side of the hemisphere encodes the experience on the right side. So he's basically reporting the right side of top of the visual field starts to move, like vibrating or moving, and without any prompt. He just spontaneously reported that. And then this is actually quite striking to me, that now he's imagining an object, okay? And then he was stimulated while imagining.
Is it vibrating? Yes.
So what he said was that he's imagining some kind of object, like an apple or whatever, and then during the stimulation, he felt that the apple was, kind of vibrating.
What about now? What about now? Remember?
So when the stimulation was either very weak or sham, then his report was also not that consistent with this experience of the moving motion of the imagined object. So this is quite striking, but that's also consistent with the idea that MT experiment in the monkey was more or less replicated in the human. So to summarize this part, area MT or B5 and the neurons in that area are likely to be causally responsible for qualia of visual motion. And the bilateral region of Avaricious motion qualia, that's called akinematopsia. And phenomenology, like motion after effects, and single neuron response, and also fMRI response, ECG response, and electrical brain stimulation, EBS, all converge to this idea that our sense of the visual motion is generated by this very, very small and particular area called bilateral MT. It's so tiny that it's very weird, but that seems to be the truth. So far, is there any question about this? Otherwise, so then the question becomes that why does this MT, this tiny area on both sides, and also one for one area, one side, and the other for the other side of the visual field, responsible or generate or correlate or causally completely, one-to-one mapping with motion qualia? That's the question. If you have any ideas, just let me know. But is there anything special about MT? Can there be any kind of a special thing here that is related to the motion? For sure, the neuron here is not moving at all. Okay, it's always static. And they have the dendrites, and they have axons, and the number of the connections in this area is more or less the same, like 1,000 to 10,000 per neuron, and it also gives an axon output. So far, nobody knows whether there is any particular molecular cellular difference in the neurons in MT that is responsible for this generation of the motion sensation. But then what the other possible kind of explanation exists is that one possibility is that the relationship or connectivity to the other areas, and another one is that within MT somehow the connection between the neurons may be different. Okay, so the question is visual motion lateralized? Yes. So yeah, just to make sure here, for example, when you see this activation here and also electrical stimulation here, it's the left brain, okay, for the subject B. And so all these response is stronger and also more specific when the stimulus is on the one side. But here, in this particular experiment, stimulation was not lateralized because this was done after this fMRI analysis was done. But as you saw in the video, when the subject was stimulated between this 1, 2, then he reported seeing the stimulus of the motion on the top right. So that corresponds to left hemisphere MT is responsible for things on the right side. Make sense? Okay, hope it's okay. All right, then moving on. So to just come back. So the local circuitry of the MT may be different from other areas of the brain, and that could be potentially related to this specific experience of the visual motion. But this link between the neural activity or connection to particular experience is a form of the hard problem. You know, nobody right now has a direct answer why this has to be the case. Why B4, B8 corresponds to the color, why MT corresponds to the motion, we don't know at this point. But before getting stuck, let's see how we can collect more evidence with a coherent research program called the Neural Correlates of Consciousness after the break in 4 minutes.